Welcome back to the deep dive. Today we're traveling way, way down, right to the molecular level,
hmm
to uncover some, well, really profound secrets in biology. You pick up a genetics textbook, and often you jump straight into the complex stuff, human chromosomes, intricate inheritance patterns.
Right, the big picture.
Exactly. But our stack of sources today reveals this fantastic twist. The biggest, most fundamental breakthroughs actually came from studying the smallest, simplest life forms out there.
That's absolutely right. And our mission today really is to give credit where it's due. We're focusing on the true architectural pioneers of molecular biology,
Yeah.
bacteria and viruses.
Simple but essential.
They aren't complex, not like us, but they were the tools absolutely central to defining the very nature of genetic material, proving DNA was the blueprint, and establishing the basic principles of inheritance that govern all life, including ours.
So we're diving into why these simple systems were, so uniquely suited for discovery,
Yeah.
how bacteria managed to share genetic material laterally, which is just key to their rapid evolution.
It's amazing stuff.
It really is. And then we're shifting gears to look at the, well, highly sophisticated ways viruses maintain control over a host cell, and, crucially, how they evade complex immune systems, like ours.
It's a real study in efficiency and cunning, actually. Okay, so to understand why bacteria are basically the cornerstone of early genetics research, you have to think about what a scientist needs most.
Speed, clarity.
Speed and clarity, exactly. If you're trying to prove, say, that DNA carries genetic information, you need a system that minimizes noise. You need something that delivers unambiguous results and fast.
We've talked before about how important that fast turnover is for research. You just can't wait 20 years to see the results of an inheritance experiment.
Precisely. And bacteria provide that speed. They're tiny, they have simple physiology, and most critically, a rapid reproduction rate. We're talking about, what, a new generation every 20 or 30 minutes under good conditions.
Yeah.
So you can observe evolutionary change in inheritance patterns basically in a single day. That's just impossible with, multicellular organisms.
Okay, but if speed was the only thing, why not just study fast-growing yeast or fungi? What did bacteria specifically offer that made them the right tool for definitive proof?
It really came down to genetic manageability
Uh,
and selection, because their entire genome is usually in one simple chromosome.
just one copy of most genes?
Generally, yeah. So it's very easy to track mutations. Plus, they display incredible genetic variability naturally. And crucially, we can easily exploit that variability using selective media.
Selective media. You mean like antibiotics,
Yeah.
where you set things up so only specific strains can survive?
Yes, exactly. If a bacterium picks up a gene that gives it antibiotic resistance that's an active allele, it lives on the antibiotic plate.
Mm.
Simple.
And if it doesn't?
It dies. It creates this beautifully clean, almost black and white readout of whether a gene is present and actually working.
Right.
And this approach was essential for experiments like the Avery McLeod-McCarty work.
Mm.
The ones who showed DNA, not protein, was the transforming principle carrying traits.
Oh, okay.
That ability to select, to track genes under pressure, it made bacteria the perfect template for figuring out how DNA actually functioned, right down at the molecular level.
That brings us to one of the most remarkable things about bacteria. They mostly reproduce by cloning, right? Just dividing.
Binary fission?
But they've evolved these amazing mechanisms to share genes laterally across individuals, which lets them adapt with, well, breathtaking speed.
Absolutely essential for their survival, and is how things like antibiotic resistance can just sweep through a whole population so quickly.
So our sources define this gene-sharing capability using three specific terms: parasexual processes.
Yeah, that's the term, yes.
Mm.
They're crucial because they explain how a population can get genetically robust, basically,
Right.
without waiting for that slow vertical inheritance from parent to offspring.
Okay, so let's unpack these. What are the mechanisms that don't need a virus to help out?
All right, first up is transformation. Think of this one as scavenging.
Scavenge.
Yeah, it's when a bacterial cell just takes up fragments of naked DNA, genetic material that's just floating around in the environment, maybe from a dead cell that burst open.
Yeah.
And then it integrates that foreign DNA into its own genome. It literally transforms its genetic makeup by picking up these free-floating bits and pieces.
Seems incredibly vulnerable, just grabbing random DNA, but also highly opportunistic, I guess.
It is. Now, what about the method that involves direct contact?
Ah.
That is conjugation. This is often described as a kind of bacterial mating, though it's not true sexual reproduction.
Right.
It needs direct physical contact between a donor cell and a recipient cell. Usually involves a specialized little tube-like structure called a pilus.
Like a bridge?
Sort of, yeah. The donor cell then pumps a copy of some genetic material, often it's a plasmid, small circle of DNA, directly into the recipient. It's like a high-efficiency genetic pipeline.
Yeah.
Great for quickly transferring things like resistance factors.
Okay, so scavenging, that's transformation. Direct pumping through contact, that's conjugation. And the third process, this is where the viruses we're about to discuss step in as like accidental mediators.
Exactly, that's transduction. This
Yep.
is gene transfer mediated by a bacteriophage, a virus that specifically infects bacteria.
Okay.
The phage acts like an unwitting courier, really. It accidentally picks up a small piece of the host bacterium's DNA while it's making copies of itself.
Oh, like a packaging error.
Precisely. And then it carries that piece of bacterial DNA along and accidentally injects it into the next bacterium it infects.
Wow. That mechanism really shows how tightly linked bacteria and viruses are, doesn't it?
Right.
Constantly exchanging information, whether it leads to, rapid evolution or just rapid death.
Exactly. Understanding these three processes, transformation, conjugation, and transduction, it's absolutely essential to grasping the sheer adaptability of microbial communities and why they can sometimes bypass our medical defenses so fast.
Okay, so bacteriophages, or phages, they're central to figuring out genetic mechanisms. Their activity is often tracked by looking for plaques on a bacterial culture. What exactly are we seeing when we see a plaque?
White plaques. They're these clear, usually circular spots you see on a petri dish that's otherwise covered by a dense, cloudy layer of bacteria, like a lawn of bacteria.
And the clear spot is...
That's where the phage has landed, infected, and killed the bacteria in that specific area. Wipe them out. Seeing these plaques form, often really quickly, was the key visual confirmation researchers needed that something was killing the bacteria.
And phages didn't just show us how bacteria died. They were absolutely instrumental in proving DNA was the genetic material itself. I'm thinking of the Hershey-Chase experiment.
The blender experiment, yes.
Right. They used phages to separate the protein part from the DNA part.
That experiment was so elegant and definitive. Phages are basically just simple structures. A protein shell protecting a core of DNA or RNA. Hershey and Chase tagged the protein coat with one radioactive isotope and the DNA core with another. Then they let the phages infect bacteria.
So spun them in a blender.
Right, to knock the phage shells off the bacteria. And they found that only the radioactive DNA had actually entered the bacterial cells to program the production of new viruses. The protein stayed outside. It basically settled the DNA versus protein question for good.
Amazing. Okay, let's use two classic examples to define the different life cycles these viral genetic masters use. Let's start with bacteriophage T4. You called it the quick killer.
Yeah, T4 is the classic example of a virus that only uses the lytic phase.
Yeah.
It's basically a smash and grab operation.
Okay.
T4 has a relatively large and complex genome, lots of genes. This lets it hijack the cell's machinery almost immediately upon infection.
Thanks, chart.
Completely. It replicates very rapidly, forces the host cell to churn out hundreds of new viral particles, and then it destroys the cell membrane, bursts it open to release all the new phages so they can go infect others. It's a simple, aggressive pathway, great for quick genetic experiments because the results are fast and dramatic.
Okay, quick and deadly. But then we have the more, let's say, sophisticated operator, bacteriophage lambda.
Lambda, yes, the sleeper agent. It has a smaller genome than T4, or fewer genes, but it demonstrates far more sophisticated genetic regulation because it has a choice.
A choice.
It can be a litigate, just like T4, destroying the cell quickly,
Quite.
or it can enter the lysogenic pathway.
Okay, detail that lysogenic pathway for us. This seems to be where the virus really takes master control over the host cell's long-term fate.
It is. In the lysogenic pathway, the lambda virus chromosome doesn't immediately start making copies.
Yeah.
Instead, it inserts itself directly into the host bacterium's own chromosome.
Whoa, it splices itself in.
Exactly. It integrates. And in this integrated dormant state, the viral DNA is called a prophage. Now, here's the key regulatory insight. The virus uses its own control system. It produces a specific repressor protein that actively keeps all its lytic genes, the killer genes, switched off.
So it silences itself?
Precisely. The virus doesn't replicate independently. Instead, it just hitches a ride. Every single time the host bacterium divides, it silently replicates the prophage DNA right along with its own DNA.
Turning the host into a stable carrier forever.
Potentially. It just passes the integrated virus down generation after generation. Until some kind of environmental stress happens, maybe UV light, maybe certain chemicals. That stress can trigger a switch, turning off the repressor, activating the lytic genes.
And then boom, it pops out and destroys the cell.
And boom. It reverts to the destructive lytic phase. It's an incredible example of genetic control and patience.
That prophage state, a piece of non-host DNA just quietly integrating itself, exercising this master control, that's the perfect conceptual bridge, isn't it?
How so?
Well, if viruses can achieve such sophisticated genetic control in a simple bacterium, imagine the complexity required to persist inside a complex host, like a human, with a vigilant, adaptive immune system constantly patrolling. Here's where it gets, I think, really fascinating.
Absolutely. The jump from a bacterial host, which has its defenses but nothing like ours, to a mammalian host, that necessitates a whole new arsenal of evasion strategies. Survival now requires actively tricking or even disabling the host's immune system.
So what are the main tricks? Our sources lay out 4 key strategies.
That's right. We can identify 4 major approaches viruses use to persist and spread despite our defenses.
What's the most fundamental strategy? The one that makes, say, the annual flu season such a relentless challenge?
That would be antigenic variation.
Hmm.
Antigens are the surface proteins on the virus that our immune system learns to recognize, like a face or a fingerprint. If the virus constantly changes these markers, mutates them, the immune system basically has to start from scratch every time it encounters the new version. It doesn't recognize the new face.
Like the flu virus constantly changing its coat.
Exactly. Influenza viruses are notorious for frequently mutating their key surface proteins, hemagglutinin and neuraminidase, the H and N at H1N1, for example.
Yeah.
It's like the virus is constantly rolling a new set of identity papers every few months, ensuring that our existing immune memory from past infections or vaccines becomes, well, less effective or even useless.
Which is why we need a new flu vaccine pretty much every year. Okay, that's variation. The second strategy seems to leverage that dormancy idea we discussed with the prophage state.
Precisely. It's latency. Some viruses have evolved the ability to enter a dormant, non-replicating state within specific host cells.
Hiding out.
Hiding out, exactly. They produce virtually no new viral particles, no new antigens for the immune system to spot. Because there's essentially no active viral presence to detect, the immune system, which relies on surveillance, simply can't find and attack it.
Can you give an example?
The classic example is herpes simplex virus, HSV. The virus that causes cold sores or genital herpes. It retreats into your nerve cells.
Nerve cells, wow.
Yeah, and it can remain silent there for weeks, months, even decades, only reactivating later, often under conditions of stress for the host. During that dormant period, it's effectively invisible to the immune system. Our defenses are robust, but they can't constantly patrol every single nerve fiber, indefinitely looking for something that isn't active.
Okay, so changing appearance and hiding. The third strategy sounds more aggressive, direct sabotage, disrupting the immune response itself.
That's inhibition of immune responses. Here, viruses actually produce specific proteins that are designed to interfere directly with key steps in the host's immune cascade. They throw a wrench in the works.
How does that work?
Take HIV, for instance. Human immunodeficiency virus. It produces proteins that specifically target and inhibit the function of critical immune cells, particularly helper T cells.
Which are like the commanders of the immune response.
Exactly. They coordinate the whole adaptive response. By crippling the commanders, HIV effectively weakens the entire defensive line, allowing the virus itself, and often other opportunistic infections, to persist and spread.
Nasty. Okay, variation, latency, inhibition. And finally, the sneakiest trick of all, maybe, fooling the host into thinking the virus is actually part of self.
That's mimicry, molecular mimicry. Certain viruses have evolved to produce proteins that structurally resemble the host's own molecules.
Like camouflage.
Like perfect camouflage, yes. If the virus successfully produces a protein that looks enough like a normal host protein, it essentially puts on a disguise. The immune system patrols, sees this protein, and thinks, oh, that's one of ours, that's self.
So it just sails past security.
It allows the viral particle to slip past detection, maybe gain entry into cells more easily, without triggering those initial alarm bells of the immune system. It's a constant evolutionary arms race, really, where the virus is always trying to stay one step ahead in molecular forgery. Hashtag tag outro.
Wow. What a profound look at the genetic world of the minuscule. So bacteria gave us the foundational template, didn't they? The speed, the simplicity needed to actually establish DNA as the stuff of genes.
They were the perfect test bed.
And then viruses came along and demonstrated this incredible spectrum of genetic control, from the, quick destructive efficiency of the lytic cycle
T4.
to the subtle long-term integration and control needed for latency and evasion, like lambda and the strategies we just discussed.
So what does this all mean? Looking at the big picture, for me, perhaps the most powerful takeaway concept is that idea of integration.
From the prophage.
Yes, the prophage showed us, fundamentally, that a piece of foreign genetic material could splice itself into a host genome, actively turn off its own destructive potential, and become a silent, integral part of that host's inheritance. It changes the host without killing it.
Yeah, that is profound. It really encourages you to reflect on that persistence, doesn't it? I mean, if viruses can exist entirely dormant inside their hosts, fundamentally shaping the host's fate and physiology over time, what other unexpected genetic partnerships might be hiding in plain sight, even in complex organisms like us?
Indeed. And it forces us to look inward, too. We actually know now that significant portions of the human genome itself are derived from ancient viral integrations, what we call endogenous retroviruses, or ERVs,
Ancient viruses in our DNA.
relics of past infections that became permanent. The sophistication of something like lambda, the sleeper agent phage, hiding out in a simple bacterium, it really highlights this huge unanswered question. How much of our own genetic makeup, our own physiology, maybe even aspects of our immune regulation, is actually defined by these quiet, dormant, viral partnerships, these things that integrated themselves millions of years ago and are now just part of us?
A lot to mull over there. That's it for this deep dive into how bacteria and viruses truly rewrote the book on genetics.
Welcome back to the Deep Dive. We're kicking off today with, well, a really fascinating biological puzzle, wouldn't you say?
Absolutely. Think about this. Bacteria, they are just phenomenal at evolving super quickly.
Right. They swap traits, they get resistant to antibiotics, adapt like crazy, much faster than, say, us.
Yeah, way faster. And here's the paradox. They do all this without the whole complex business of sexual reproduction that we rely on.
No meiosis.
Yeah.
No, what's it called? Chiasmata?
Exactly. No chiasmata, that physical crossing over of chromosomes that mixes everything up for us. Bacteria just bypass all that.
So how do they do it? They must have some tricks up their sleeve.
They do. Instead of that slow shuffle of genes and sex, they use three incredibly fast, sort of targeted shortcuts. And crucially, it's always one-way traffic.
Okay, so that's our mission for this deep dive then. We're looking into these three parasexual processes.
Right.
Transformation, transduction, and conjugation.
Mm-hmm. And these aren't just minor details. They're the key mechanisms behind things like rapid antibiotic resistance spread.
The shortcuts that let a bacterium suddenly survive a new drug or, I don't know, digest something new.
Precisely. So before we get into the how, we should probably lay out the basic genetic tools bacteria work with. Think of it like their DNA toolkit.
All right, what's in the box?
Well, first, you've got the main player, the big circular bacterial chromosome, several million base pairs, holding all of the essential genes for just, being a bacterium.
The core operating system.
Exactly. Then you have these smaller auxiliary pieces. Plasmids are a big one.
Oh, yes, plasmids, little circles of DNA.
Tiny circles, yeah. They replicate on their own, separate from the chromosome. They're not essential for basic life, but they carry, like, bonus genes.
Like the R plasmids. For resistance.
That's the most famous example. Our plasmids are major carriers of antibiotic resistance genes. Huge survival advantage right there.
Okay, chromosome, plasmids. What else?
There's a third type, sort of in between, episomes. These are also circular DNA, generally bigger than plasmids and also non-essential.
So what makes them different from plasmids then?
Their flexibility. An episome can hang out on its own, replicating like a plasmid, or it can actually integrate itself, slot right into the main bacterial chromosome.
Oh, interesting. So it can be separate or become part of the main genome.
Right, and the classic example is the F factor, the fertility factor. We'll definitely come back to that one.
Gotcha. So chromosome, plasmid, episome, and the key rule for transfer.
Always unidirectional, always donor to recipient, no back and forth.
Okay, crystal clear. So let's get into the three mechanisms. How do scientists tell them apart? You mentioned things like cell contact.
Yeah, two main questions usually help distinguish them. First, do the bacteria need to physically touch for the transfer to happen?
Okay.
And second, can the process be stopped by adding an enzyme called deoxyribonuclease, or DNase, to the surroundings?
DNase, that chews up DNA, right?
Exactly. It breaks down naked DNA that's just floating around. So if DNase stops the transfer, it tells you something important.
All right, let's start with the first one. Transformation. What's the deal there?
Transformation is maybe the simplest. Think of it as, well, scavenging.
Scavenging, like picking up leftovers.
Kind of, yeah. It's the uptake of free DNA that's just out there in the environment. Maybe another bacterium died and burst open, releasing its DNA fragments.
So a living bacterium can just Slurp up that free-floating DNA.
Pretty much, but there's a condition. The recipient cell has to be in a special state called competent.
Competent, meaning what?
It means it's switched on the specific cellular machinery needed to bind to that external DNA and pull it inside. It's not always on.
Okay, so it has to be ready for it, and once it's inside.
Once inside, that fragment can potentially recombine with the recipient's chromosome, swapping out a piece of its own DNA for the new piece. If that happens, the new trait is stably inherited.
And this is where that DA's question comes in, I bet.
Exactly, because the DNA starts out outside the cell, just floating free.
DNA's in the environment would chew it up before it could even be picked up.
Precisely. So transformation is uniquely sensitive to DNase. That's a key diagnostic test.
Makes sense. Now, you mentioned fragments. How much DNA actually gets transferred this way?
It's actually a tiny amount, usually only about, say, 0.2 to 0.5% of the entire bacterial chromosome in one go. Very small pieces.
Less than 1%. Wow. Okay, why is that detail useful? Seems limiting.
But it's incredibly useful for mapping genes. Because the fragments are so small, if two genes get transferred together frequently...
It must mean they were physically very close together on the original DNA fragment that got picked up.
Exactly. It's called co-transformation frequency. If gene A and gene B always come across together, they must be right next to each other on the chromosome. It tells us about linkage.
Using random chance to figure out the gene order. Clever.
It's a classic technique.
Okay, so transformation is scavenging DNA scraps. What about conjugation? You mentioned this might be like bacterial sex.
Yeah, it's often called that because it involves direct cell-to-cell contact. It's a much more deliberate transfer.
How does that work?
Yeah.
They shake hands.
Sort of. One cell, the donor, actually builds a physical bridge or channel, it's called a pilus, that connects it directly to the recipient cell.
A bridge between cells. Wow. And what allows a cell to do that?
That's where the F factor comes in the fertility episode we mentioned. If a cell has the F factor, it can usually build a pilus and initiate conjugation. We call it an F+ cell.
And the recipient would be F, liking the factor.
Typically, yes. An F+ cell connects to an F cell.
Okay, physical contact. How did researchers prove that contact was absolutely required? Couldn't the DNA just leak out nearby?
The famous YouTube experiment. Classic stuff.
Tell me about it.
They put two different bacterial strains in the two arms of a U-shaped tube. Separating them at the bottom was a filter.
What kind of filter?
One with pores so fine that liquids and dissolved stuff like free DNA could pass through, but the bacteria themselves couldn't cross.
So they could share the same broth but couldn't touch.
Exactly. And under those conditions, zero gene transfer between the strains. It proved you absolutely need that physical connection, the pilus.
Brilliant. Okay, so the bridge is formed. How does the DNA actually move across?
It usually uses a process called rolling circle replication.
Rolling circle.
Yeah. Imagine the circular F factor, or sometimes even the whole chromosome, if the F factor is integrated. One strand gets nicked.
Nicks, like cut.
Just a single strand cut. Then that nicked strand starts to unravel, and as it does, it gets fed through the pilus into the recipient cell.
Like pulling thread off a spool.
Exactly like that. And as the old strand spools off into the recipient, the donor synthesizes a new strand to replace it, keeping its own DNA double-stranded. The recipient also synthesizes a complementary strand for the DNA it receives.
So both end up with a copy. Very efficient.
Extremely. And this mechanism becomes incredibly powerful for mapping genes, especially using what's called an HFR strain.
HFR, what's that?
High frequency of recombination. This is a special type of donor cell where the F factor isn't separate anymore. It has actually inserted itself into the main bacterial chromosome.
Like you said episomes can do.
Right. So now when this HFR cell starts conjugation, it begins transferring the F factor DNA, but because it's stuck in the chromosome, it starts dragging the entire chromosome along behind it, bit by bit, through the pilus.
The whole chromosome?
Right.
Wow, does it usually transfer the whole thing?
Rarely. The connection is fragile and usually breaks before the whole thing gets across, but the key is the chromosome genes enter sequentially in a fixed order, starting from where the F factor inserted.
Sequentially, like beads on a string being pulled through.
Perfect analogy. And because it's sequential.
You can use time. You can measure how long it takes for specific genes to arrive.
Bingo. That's the conjugation interruption experiment. You mix HFR donors and F recipients, let them start mating, and then at different time points, say every minute you violently disrupt them.
Like in a blender, seriously.
Literally, yes. A kitchen blender works perfectly to shear the connections. Then you test the recipients to see which genes made it across before the interruption.
So time equals distance on the chromosome map.
Exactly. The standard map unit in E. coli genetics is 1 minute of transfer time. The entire E. coli chromosome map is about 100 minutes, or 100 map units.
That's amazing. Can you give an example with actual genes?
Sure. Let's say you do this experiment. You might find the TRAP plus gene for making tryptophan gets transferred after about 7 minutes.
Okay, 7 map units in.
Right. Then maybe the TIR plus gene comes across at 23 minutes.
Further down the line.
Keep going. Maybe an important antibiotic resistance gene, ABR, shows up at 31 minutes, and then maybe ARG plus at 42 minutes, LU plus at 49 minutes.
So you build up the exact order and relative distances just by timing it.
Precisely. It's a beautiful, relatively low-tech way to create a detailed genetic map.
Incredible. Okay, so we've had scavenging, transformation, and direct bridging. Conjugation. What's the third way? Transduction.
Transduction involves a middleman, a bacteriophage.
A phage. That's a virus that infects bacteria, right?
Exactly. Phages hijack the bacterium's own machinery to make more copies of themselves.
Sounds nasty for the bacterium.
It usually is. But sometimes during this replication and assembly process inside the host cell, things get a bit messy.
Messy how?
When the new phage particles are being assembled, sometimes completely by accident, a piece of the host bacterium's chromosome gets packaged inside the phage head instead of, or sometimes along with, the phage's own DNA.
Whoops. So the phage is carrying bacterial DNA now.
Yeah, it's like a mispackaged parcel. Then when this faulty phage goes and infects a new bacterial cell,
It injects the DNA it's carrying, but instead of just viral DNA, it accidentally injects the piece of bacterial chromosome from the previous host.
exactly. And that piece of DNA can then recombine into the new host's chromosome, transferring the trait. The phage acted as an unwitting delivery vehicle.
And since the DNA is protected inside the phage particle until injection,
It's not exposed to the environment, so like conjugation, transduction is not sensitive to DNase.
okay, so 3 distinct mechanisms. Scavenging free DNA, building a bridge, or getting carried by a virus. Why is this whole system so important? What's the big payoff for bacteria?
The payoff is speed and adaptability. These mechanisms let bacteria acquire potentially life-saving traits, like resistance or new metabolic pathways almost instantly from their neighbors, without waiting for slow, random mutation and generational inheritance.
They're sharing survival strategies in real time.
Absolutely. It's horizontal gene transfer, a massive driver of bacterial evolution, especially in response to environmental pressures like antibiotics.
And we can actually see the results of this genetic shuffling, right? And it's not just invisible DNA changes. It shows up in their characteristics, their phenotypes.
Oh, definitely. Scientists rely on these observable changes. Think about colony appearance.
Like color.
Yeah, a great example is Serratia marcescens. Normally, it makes this bright red pigment, prodigiosin. But if a gene in that pathway gets mutated or changed via one of these transfer methods, the colonies suddenly appear white.
So the color change signals a successful gene transfer event.
It's a very useful visual marker. Or think about what they can eat, their nutritional needs.
Right, you can get mutants that suddenly can't make something they need.
Exactly. This gives us the key distinction between prototrophs and auxotrophs.
Prototrophs are the standard wild type ones.
Yes. Prototrophs can grow on minimal medium. They can synthesize all the essential amino acids, vitamins, etc. that they need from basic ingredients.
In auxotroph.
Auxotrophs are mutants. They've lost the ability to make one or more essential nutrients because a gene got knocked out or changed. So an arginine auxotroph, for instance, needs arginine added to its growth medium to survive.
And this difference is how scientists track the transfers. Like seeing if an auxotroph recipient gets rescued by DNA from a prototroph donor.
Precisely. Selecting for the gain of function, turning an auxotroph back into a prototroph, is fundamental to detecting and quantifying transformation, conjugation, or transduction events.
And of course, the phenotype we keep coming back to, antibiotic resistance.
Yes, arguably the most impactful consequence in the human world. Gaining resistance through acquiring an R plasmid via conjugation or a resistance gene via transduction is a direct, observable, and often dangerous outcome of these processes.
So let's wrap this up. We've seen these 3 ingenious, if somewhat accidental, ways bacteria share genetic information.
Right. You've got transformation picking up naked DNA scraps, needs competence, stopped by DNase.
Then conjugation, The direct cell-to-cell transfer via pilus, driven by the F factor, great for mapping using time, not stopped by DNase.
And transduction, the phage delivery service, accidentally packaging and transferring bacterial genes, also not stopped by DNase.
It really is an incredibly efficient system for them, especially when you think back to that conjugation mapping.
Yeah, where acquiring a vital gene might be separated by just a few minutes of contact time.
It makes you think, doesn't it? If bacteria have perfected this incredibly rapid, one-way sharing of survival information resistance, metabolism, you name it, measured in minutes, what does that really mean for us, trying to control them?
It means we're up against an opponent that shares winning strategies almost instantaneously across populations. Their ability to transfer information horizontally is perhaps their greatest evolutionary weapon in the ongoing arms race.